So we are turning to a different topic now, which is prevention of diabetes, and I'm going to discuss the issue of fatty acids in the development and prevention of diabetes. I'm going to talk about the Finnish Diabetes Prevention Study that we carried out in the people with impaired glucose tolerance who were obese and middle-aged. We randomized them into a lifestyle intervention group and the control group, which control group received very basic advice for healthy lifestyle. And the intervention group received management by dietitian and physical activity experts. So the aims of the intervention were very simple: weight reduction, change in diet, basically concentrating on the fat intake, reduction of fat intake, especially saturated fats, increase in fiber intake, and increase in physical activity. So nothing very extraordinary. But this program we had resulted in favorable changes in dietary patterns, as shown here. The fat intake was changing, saturated fat intake especially, which was the aim. And dietary fiber intake was increasing. So this is kind of the intervention program that can be carried out in any of the routine setting. The program resulted in 58% reduction in the development of diabetes over the years. On average, we had about 4-year intervention, and you can see very clearly that the intervention worked very well, and it started to work already at the very early stages of the program. Now, after the program was over, and we had evaluated the efficacy of the study. As I showed you, 58% reduction in the risk of diabetes. We continued the follow-up of these individuals then for another 10 years with the annual examinations in order to see who developed diabetes and who remained non-diabetic. So here you see that after stopping the intervention, we still observed a clear difference between the groups. So there was no relapse in the efficacy. So the 4-year intervention actually carried out the benefits up to 15 years. That was our latest examination year in 2009. So this was a very good news that the intervention actually is lasting much longer than it was carried out. And during this study, we could see that especially fat intake in the intervention group was reducing dramatically, saturated fat intake as well, fiber intake was increasing. So the benefits were there very clearly. And even after stopping the intervention, we still had a clear difference between the intervention and the control groups in the fat intake, saturated fat intake, and fiber intake. So this was the basics what we saw. We know that dietary fat intake is associated with the development of diabetes, and it happened in our study as well very nicely. that in those individuals who had the highest fat intake actually had the highest incidence of diabetes. And it could be seen also in the saturated fat intake here. So obviously there is something going on with the fat intake and diabetes. On the other hand, we can see that also the dietary changes are resulting in changes in serum lipids, especially in this case the increase in HDL cholesterol and decrease in triglycerides, because these are of course the components that are usually disturbed in the people with impaired glucose tolerance and diabetes. Now the biomarkers may be better indicators of dietary fat intake than the simple dietary questionnaires or of food diaries. We use the food diaries that are pretty good, but obviously the objective measurement of biomarkers may be even better. And we know that also there are different genetic variations that are associated with the fatty acid composition in the serum. Now the measurement is not very simple. You need the chromatography and so on. The most viable markers are those that are related to marine products, fish oil, and these can be measured either from erythrocytes or adipose tissue or from serum. And also we can measure the enzyme activity that is linked with the dietary fatty acids, this delta 6, delta 5, delta 6, delta 9 desaturase enzyme activities. And what we know about the fat quality and the development of diabetes is that there are some indications that these delta 5, delta 6, delta 9 desaturases and several fatty acids could be related to type 2 diabetes. But when we look at the data published related to, for instance, omega-3 fatty acids and fish intake, the results are really conflicting, and it's not really known whether these omega-3 fatty acids would be protective against type 2 diabetes as they may be against cardiovascular disease. So in our study, we took our serum samples and investigated the serum fatty acid concentrations. We had about 400 individuals for whom we had samples available, and we used the 11-year follow-up for this purpose. And we also had a possibility to look at the different insulin sensitivity indices, so-called Matsuda index or insulin secretion index from fasting to 30-minute insulin secretion and the glucose levels. So we were able to analyze the data in a very detailed way. So we had the samples for several annual examinations that we could use, and then using statistics to control for different confounding factors, we could analyze our data. And the assays we did with a company in California, Tethys, that was a company that was collaborating with us, and we could to measure the fatty acids in our samples with them. The main fatty acids we used in the analysis are listed here, so they are alpha-linoleic, arachidonic acid, and then the DHA, DPA, and then the delta-5,6 desaturase and EPA, linoleic acid, sterol coenzyme A desaturase. So we tried to see which one of these actually worked in the development of diabetes, or which were correlated in insulin sensitivity or insulin resistance, if you want the other way around. And here you see the taking the model where we had a multivariate adjusted with the various things with the diet, body mass index, waist circumference, physical activity, etc. And you see here that there are, on red, the significant fatty acids. There were several of them that were associated with insulin sensitivity. And also several of the fatty acids were in the unadjusted analysis associated with the development of type 2 diabetes in our cohort, you see here, but actually there were only 4 that were associated after adjusting for confounding factors. So the arachidonic acid and EPA, DPA, DHA were actually the significant parameters. So here you see the example, for instance, the EPA so there was a protection in the highest level compared with the lowest level of the EPA. So obviously some marine fatty acids were protective. Similar here for DPA and again for DHA. So all these 3 fatty acids were looked like a protective factors, and then the enzyme activity, delta-5 desaturase activity again, if it was low, was associated with the higher progression to diabetes. Another measurement we did was sterol measurements. There are so-called plant sterols, beta-sitosterol and campesterol, that are reflecting a cholesterol absorption. And then also the markers of cholesterol synthesis, latosterol or desmosterol, and these were measured in the same methodology with the same company that teaches bioscience. So we could actually look at the cholesterol absorption, cholesterol synthesis, whether they were correlated with the risk of diabetes. Here the first is to look at the correlations with the body mass index or acute insulin response to glucose. And you see that there were significant correlations with the beta-sitosterol, campesterol, and latosterol, but the latosterol actually had the opposite effect compared with these others. So if there were positive findings here, it was negative in latosterol, or vice versa. If it were negative for the acute insulin response, so it was actually increased in latosterol. And when we looked at the plant sterols and latosterol in the intervention and the control group, in the plant sterols, the intervention group actually had increased levels of both beta-sitosterol and campesterol, but no difference really in the latosterol, reflecting the lifestyle intervention. Another interesting finding we had, because we had a genotyping done in a very large scale, genome-wide genotyping of our study. We were able to look at the different genes, and this is one of the APC G8 SNP, which is known to be associated with the cholesterol or lipid levels in general. And what we could see that individuals who were carriers of a G allele, in the intervention group, they actually had higher levels of plant sterols, both beta-sitosterol and campesterol. So there was a clear effect, but those who were the minor allele T homozygous had no effect on these sterol levels. So quite interesting that the lifestyle intervention had these effects, and we know that the beta-sitosterol and campesterol, they are associated with the control of high LDL cholesterol levels in general, and these are used in the food additives in the functional foods nowadays. And to look at the 8-year risk of diabetes, see that in beta-sitosterol and campesterol, there were significant reductions, about 20-25%, even after controlling for body mass index or insulin sensitivity index, or the acute insulin glucose ratio. And the latosterol had the opposite effect, so that there was an increase about 50% in the one standard deviation change. So the plant sterols seem to be associated also for the risk of type 2 diabetes. When we looked at this APC-G8 SNP, it had no significant effect on the risk of diabetes or effect on plant sterols on the type 2 diabetes. But however, as I showed you, it had an effect on the plant sterol levels. So in the conclusion, these marine omega-3 fatty acids, EPA, DPA, and DHA, and estimated delta-5 desaturase activity are predicting type 2 diabetes incidence, and probably they are improving insulin sensitivity. And the question is whether fatty efficient intake actually is protective against type 2 diabetes when measured in a proper way. As I said earlier, the fish intake per se seems in many studies not be associated with type 2 diabetes protection, but in our case, when we measure the fatty acid composition in the serum, there seem to be effect. Also, this the enzyme activities, delta-5 desaturase activities, it's really not known how it works, but it could be that it has a regulation of a production of arachidonic acid, which was also protective against type 2 diabetes in our study. And then plant sterols that are reflecting cholesterol absorption are inversely associated with type 2 diabetes, whereas increased cholesterol synthesis indicated by latosterol levels are associated with a higher incidence of type 2 diabetes. And the levels of plant sterols, beta-sitosterol and campesterol, are controlled to some extent by the gene APC G8, which is associated with the lipid levels in the population level. So my colleagues here who have been working with this study are shown here. These data are being published in the near future in the 2 papers that have been accepted for publication. So thank you very much. And now I will ask the questions.
How much do you use EPA and EPA to reduce the incidence of diabetes?
Sorry?
How much dose of EPA and EPA?
No, it's not. We cannot measure the dose. It's just the level of a serum. We don't know the dose. It's just the serum level.
I know serum level, but you said that the intake of EPA and DPA can take it orally to reduce the incidence of diabetes?
There are no data on that. Nobody has really done a proper study to see the dose response. When you look at the fish intake dose response, you don't see any. But specifically, there are no studies that have been looking at the controlled trial to give a different doses of these marine fatty acids. There are no data.
But if there is such a fact, why you don't try to go through these ideas that increase the intake and see whether affect diabetes incidence?
Good question. On the other hand, I have to remember that many studies, when you give a specific chemical, like a certain vitamin, you don't see any effect. But if you look at the dietary intake with the high vitamins and so on, you see an effect when it comes together with the certain foods. So it's always a problem when you try to take out one chemical from your diet and give it to people that, well, this is a magic thing. It doesn't work that way. But if you have a healthy food, which incorporates many other things, it seems to be working.
Thank you.
Thank you very much for that. My name is Jennifer. You're talking about the lifestyle intervention. We are developing digital technology that helps facilitate that signposting, and we've carried out a study, a very short intervention, and now here in Dubai in January, a 9-month intervention. So I'm very interested in the 4-year and the 11-year tenure of this study. What are the challenges that you face in trying to retain the participants in a study as long as this?
So I didn't catch your question.
So 4-year, this is a 4-year study, leading on to an 11-year study.
Yeah.
How difficult is it to retain the participants in the study for that length of time?
Yeah, we had a dropout rate about 15% of the 11 years. So basically everybody was more or less retaining if they were healthy enough to come to the clinical examinations. And also, when we look at their lifestyle, we measure their dietary habits and physical activity. They kept their healthy lifestyle over the years, so it seemed to be working that way.
Thanks.
Okay, I think we have to go on, and first I have to give the document for myself. Thank you very much.